Hello everyone, my name is Shaira Feroz. Today I'll be presenting on persistent Ebola infections and how genomic methods gives us an insight into relapse and transmission. I'll focus on two real-world Ebola outbreaks and show how genomics and bioinformatic tools are now essential part of outbreak response. Before diving in, a little about the virus and why it's so scary. This virus, Ebola virus, is rare but extremely severe, and it is known for having a high case fatality rate, especially during outbreaks in areas with limited healthcare resources. Fruit bats are thought to be natural hosts, and transmission to humans occurs through contact with infected animals or direct human to human spread through blood and body fluids. Unfortunately, unsafe healthcare practices and traditional burial ceremonies often contribute to this kind of spread. One of the key challenges is that the virus can persist inside survivors for months after recovery, and although it's rare, this persistence can lead to dangerous flare ups and secondary outbreaks. The first case that I'll be discussing is from 2018 to 2020 North Kivu outbreak in Congo, and this was the second largest Ebola virus outbreak, which had about 3,400 cases in total. The diagram shows the timeline for a patient his and from the and from his first vaccination to his demise. So the patient was first vaccinated in December 2018, and although he initially recovered, he relapsed again 6 months later. And then a couple months later, again in June, um, after he tested positive, he had a treatment of monoclonal body, and he again got cured and was tested negative for the Ebola virus. But again, he tested positive for Ebola just 6 months later, and tragically, this time he passed away. So, this figure on the right shows the events of his association with this virus. So, soon after his death, genomic investigations were conducted, and it showed us that the virus had a very slow evolution with unique mutations emerging. Um, his relapse actually led to new transmission chains as well, causing 91 additional cases across 6 health zones. The phylogenetic tree was drawn, and it could reveal critical findings. It showed that the rate the reduced rate of the virus was about 0.2 mutations per 1000 sites per year, which is less compared to the normal 0.8. This evolution confirmed that the viral persistence was from the inside and not really from the outside. 2 additional mutations were also found during the relapse episode, allowing the scientists to trace the entire cluster back to this single relapse event. The second case comes from Uganda in 2025. On January 29, the Ministry of Health declared an outbreak caused by Sudan virus, which is a different species but of Ebola still. This is the 8th Ebola outbreak in Uganda since 2000. The patient was a nurse who showed classic Ebola symptoms from developing high fever to chest pain, difficulty in breathing, and to multiple bleeding from different body sites. He did sought treatment at multiple health facilities, but he still passed away on January 29th of this year. So let's move to the real-time genomic work. As soon as this person was um, tested positive, his blood samples were collected and sequenced by the Central Public Health Laboratory, using real-time PCR and whole genomic sequencing, they quickly confirmed that it was Sudan virus, which is one of the 6 known species of Ebola virus, and not Zaire Ebola. Zaire Ebola is the 2022 is the causative agent for 2022 outbreak, whereas the Sudan is from 2012. So this was done in like 24 hours, and the researchers actually had to camp out overnight just to finish this work. Using IQ3, efficient software for phylogenomic inference, they do phylogenetic analysis and could discover that again the 2025 strain was closely related to 2012 and not 2022. This ruled out the possibility of sustained human-to-human transmission from 2022 and suggested a natural spillover from an animal reservoir might be the cause. For the first time in Uganda's history, genome sequencing, analysis, and public reporting were all achieved within 24 hours, which is an extraordinary speed. However, again, it happened in 2025, and we need to ensure that we can monitor and predict outbreaks and ensure that, like 2020, there is no relapse of the virus. Even though the present outbreak was due to natural spillover, there had been 9 confirmed cases, and although majority has completed quarantine and been discharged, it hasn't been 6 months like the previous case, and we need to stay prepared just in case, so that we can again deal with it within 24 hours and ensure that no lives has been lost. So I heard of this fear detector from another paper, which I feel like can significantly aid in dealing with 2025 outbreak. So fear detector is like a new bioinformatics pipeline designed specifically for virus surveillance using nanopore sequencing data. It mainly has 4 outputs. First is identifies the species and its lineage, then it reconstructs the viral genome, then it detects the structural variants, and lastly it builds phylogenetic trees. In context to the 2025 outbreak, it could be invaluable, as in it can provide rapid identification, it can track transmission chains, it can detect persistent infections, and most importantly, it can inform real-time public health responses and survivor monitoring. I'll briefly go how the tool works. So first we input data here, which are raw nanopore files or FASTQ files. Then using Guppy software or Dorado, which we convert the raw electrical signals to nucleotide sequences. Then we trim it out with nanofilter to clean and remove low quality reads. Then we assemble them, which is we either use CanU if there is no reference genome present, where this CanU will assemble the viral genome from scratch, or we can use BLAST to find the closest reference genome. And using that reference genome, we map it, and followed by detecting structural changes using variant detection, and lastly followed by alignment using either MAFT or muscle, followed by phylogenetic tree made using IQ32 or Mr. Bayes. So in conclusion, Ebola is a huge concern to the world because they have the potential for outbreaks caused by relapses. Real-time genome sequencing, phylogenetic analysis, and variant detection allows us to rapidly trace these origins. And the situations that I highlighted today, particularly the 2025 one, are very recent and require applying solutions from variety of studies because comprehensive datasets are still under development. Moving forward, integrating machine learning models can be extremely critical, or we can also move towards creating dynamic hubs or real-time centralized genomic data, which can all enable to exchange and compare viral genomes across boundaries. More advanced bioinformatics tools are also required, because in doing all of these, are we able to make sure that we can fight against Ebola and other new viruses? And it's not just promising, but it's also scientifically exciting. These are my reference, and thank you. Any questions?